Diffusion, the international science radio show.
We have a bouncer on the doors of perception.
The good, the bad, the ugly.
It gets pretty exciting.
The myths, the truths.
Toxicology.
Astroseismology.
Magnetism. The dark side.
Genetically engineered potatoes.
Planetoid.
I
love that word.
Hello and welcome to Diffusion. Sit back and relax while we enculturate your brain with weird and wonderful science. I'm Ian Woolf. On this edition, voice clones, archive attacks, and from the vault, crocodiles, alligators, and an interview about embryonic stem cells. But first news of podcast problems. My podcast host has been down for weeks. I wrote some news about it and then trained the latest AI voice cloning models on my own voice using an old episode of Diffusion that included news about organoids. As you listen to the fake Ian read the news, please note that there were no organoids mentioned in the news script at all, just in the training sample. The software I used is free on GitHub as F5-TTS. And now here's the real news with a fake voice.
Here's the news.
Podcast down! The organoids are coming. The Diffusion Science Radio podcast has been down since vandals attacked the Internet Archive on the 8th of October 2024. This podcast is hosted on the Internet Archive, so if you used a podcast app, the organoids are coming. You wouldn't have been able to listen to any episodes you hadn't already downloaded. First, the organoids are coming. The only place you could hear new episodes has been the Community Radio Network and Spotify. I've put some episodes up on YouTube. The organoids are coming. The Internet Archive is the world's largest archive of the Internet. Their mission is to provide universal access to all knowledge. The organoids, they archive old websites, old software, old records, old radio shows. The organoids are coming. Old films, anything they have a copyright to share as a library for free, as a service to the public. The Internet Archive is a private non-profit organisation based in San Francisco that operates on a very low budget. It was founded by Bruce DeCarl in 1996. First, the organisation said its vast cache of archival material is safe following the breach. However, they had to take the entire website offline while they redesigned their security completely. Organoids coming. The Internet Archive have been gradually restoring services as read-only. They started with the Wayback Machine. The organoids are coming, which can show you websites archived back to the first days of the World Wide Web. The organoids are coming to government web pages that later governments tried to erase, to your old blog. They just put the complete archives, which include all the episodes of Diffusion, back online today, but read-only, so I can't add new episodes there yet. But first, the organoids are coming. The attack on the Internet Archive leaked identifying information from more than 31 million user accounts, including patron email addresses and encrypted passwords. According to the website Have I Been Punned, which tracks accounts that may be compromised in a data breach. As the passwords are encrypted, the organoids are... They can't be read by the attackers to hurt the users further. But first, the organoids are coming. They were also attacked in court in the last few years by publishers who have admitted in court that they ultimately want to shut down all public libraries. First, the organoids are coming. The Internet Archive digitized a large collection of books they had purchased, organoids are thing, and books that had been donated from other libraries. The organoids are coming, and then made single copies of the books available to read online for a limited time, by one person per physical copy of the book. The organoids are coming! They did this to help people during the long lockdowns of the beginning of the coronavirus pandemic in 2020. Despite never making any money from this, the publishers successfully argued that lending books you own is piracy. But first, the organoids are coming. Brewster Cable told the Washington Post that the costly fines from the lawsuits could end up killing the archive. By coincidence, the British Library, Seattle Public Library, Toronto Public Library, and Calgary Public Library were all recently attacked online as well. The organoids are coming. I'm looking for alternative ways to host the Diffusion podcast for free. First, the organoids are coming, so that I'm not knocked out again when the Internet Archive is next attacked by enemies of free speech. The remainder of the news and the interview that follows are from 2014.
Crocodiles in trees. Thought you were safe out of the water and off the ground? A University of Tennessee study has found that crocodiles can climb trees. The study observed crocodiles in North America, Australia, and Africa. The researchers found 4 species of crocodiles that climb trees. Smaller crocodiles could climb higher than large ones. Crocodiles were observed climbing as high as 4 metres up a tree and 5 metres down a branch. Crocodiles climb to control their temperature, to watch for threats, and of course, to catch prey. The study also mentions Australian crocodiles climbing chain-link fences up to 2 metres tall.
The study was published in Herpetology Notes and titled Climbing Behaviour in Extant Crocodilians.
The lead researcher was Vladimir Dinets, a research professor in the Department of Psychology at the University of Tennessee. Who knew psychology was so dangerous? Professor Dinets has also published observations of crocodiles using sticks to catch birds. Mugger crocodiles in India and American alligators were seen using sticks and twigs to lure birds during nest building season. The crocodiles and alligators stayed still for hours and then would suddenly lunge if a bird approached the sticks on their nose. The paper was published in Ethology, Ecology and Evolution under the title Crocodilians Use Tools for Hunting. From crocodiles with sticks to alligators with cameras, researchers strapped National Geographic critter cams onto the backs of American alligators to study their feeding habits up close. The critter cams were held on the back with a backpack-like harness around their waist. Turns out the alligators eat a lot of crayfish and turtles and mostly hunt at night. They were most successful between 4 a.m. and 9 a.m. The article appeared in National Geographic and the paper was published in PLOS, the Public Library of Science 1, titled Animal-Borne Imaging Reveals Insights into the Foraging Behaviours and Deal Activity of a Large-Bodied Apex Predator, the American Alligator. The videos will be embedded on the Diffusion Radio webpage for this show. I myself strapped a camera onto a rather smaller and friendlier animal last weekend. I taped my Luxie wearable camera to the collar of a 7-month-old Chihuahua puppy accompanied by his human on a trip to Centennial Park. You can see the video of Doggy Cam, Toby's Day Out, on the Diffusion webpage for this show. Toby is very close to the ground and he rocked the camera up and down whenever he ran, which was most of the time.
Toby failed to hold any sticks still long enough to catch birds, he didn't climb any trees, and he left alone the chain-link fences.
You're listening to Diffusion Science Radio. Send emails to science@diffusionradio.com. We're brought to you across Australia on the Community Radio Network and podcast over the internet on www.diffusionradio.com. Michael Morris is head of the Embryonic Stem Cell Laboratory at the Bosch Institute of Physiology at the School of Medicine at the University of Sydney. He spoke to me about investigating the basic science of embryology to find out how stem cells turn into other cells.
So 2 things. One is that they have the ability to turn into any cell type, so they're pluripotent. So we like to know what the molecular mechanisms are that maintain that sort of very naive state. And then you can drive them down to differentiate to particular cell types. So we want to know how to do that as well.
And so you're actually observing what happens as embryos grow.
That's right. Effectively, I mean, these are cell lines, so they grow in tissue culture, but they act as a laboratory model of how embryos grow.
It seems to me that there's so many different tissues in the body, there must be so many different ways for the cells to get instructions.
There's a surprisingly small number of instructions, but they can be used in different combinations and at different concentrations. So if you go to the adult body in humans, there's about 220 different cell types. And there's a few more of that get produced along the way as the embryo grows, but there's not a huge number of cell types. But those signals combine to drive a naive cell through all of the different cell types to get to a mature cell type like a skin cell or a nerve cell. And that's how the process really develops. Different combinations of factors working at the right time at the right place as the embryo develops, so that everything occurs in the right order.
And so what are the challenges to finding out what these signals are and the order that they need to happen in?
The challenges are finding the molecules that you can put into the system that will drive the steps sequentially. And generally they're things like growth factors, for example, and cytokines. Usually protein molecules, but also there are some small molecule growth factors that also have very important roles in the developmental process of embryos. So the starting point really often is what growth factors and cytokines you need to drive the steps sequentially. Those growth factors and cytokines generally bind to receptors and stimulate what we call cell signaling pathways, which is a cascade of events inside the cell. So really what you're doing is transferring information from outside the cell with the growth factor, binds to a receptor, and that information is transferred inside the cell and decoded. And the outcomes of that can be the differentiation process for development to proceed.
The differentiation that you're learning for all of this, what are the uses to which this technology, well, these techniques will be put?
There's two main things. The general public will know about the major one, and that is the production of particular cell types that might be used in cell therapies for a range of human diseases and injuries that are currently very poorly treatable or not treatable. So, in other words, cell therapy is what most people think about. But the second thing is really underpins that and actually is, in a sense, much more important. And that is simply learning what the molecular mechanisms are in the processes of development. So if you understand the mechanisms in sufficient detail, you can then begin to understand how normal and abnormal development proceeds. And if you know those things, then you can begin to get much better clues about how to intervene when things go wrong, or how to fix things when things have gone wrong. Now, whether that's by using a cell therapy or developing an appropriate drug or some combination of those things is another matter. But really, most of the information that's come out that's been really useful is that understanding of normal and abnormal developmental processes.
And that's something, it's an ongoing investigation. There's still a lot to understand.
A huge amount, because I talked about maybe using combinations of growth factors outside the cell. You might have 2 growth factors, but each of those might, for example, stimulate half a dozen signaling pathways each. And each of those signaling pathways interact in various complex ways. And below that, there are different genes that are then expressed or not expressed on the back of all of those signaling pathways. So it becomes an enormously complex process inside the cell. The amazing thing is the cell has the ability to decode all of that information and make the appropriate decisions. And there could be a number of decisions that the cell makes. It might be to differentiate, to grow faster, or to grow slower, to not die, to change shape, to go on the move, or any combination of those things. So it's very complex, and we are beginning to understand that we need to understand that complexity in order to have the best chance to understand what's going on, and then in order to have the best chance to intervene appropriately and trying to fix things.
So what are some of the projects you're working on at the moment?
So we're working on three main things. One is looking at the ability of these naive stem cells to make neural cells. And in order to do that, they need to go down a pathway of development which includes a number of intermediate steps. We've developed protocols for producing those cells, which have some novel growth factors in them that have not been described before. So that's interesting from our point of view. It also means that ultimately we should be able to produce cells that might be used in animal models of human diseases and injury. We're also interested in the fact that nutrition plays a big role in development because the cells need to pick up their nutrients in order to survive. But what I think we're beginning to understand is that metabolism that comes from the addition of nutrients into the medium is not just a means of keeping the cells alive, it's actually required to make the cells differentiate appropriately. So there's this synthesis between differentiation processes of cells and the metabolism of cells. The two aren't distinct from one another, they actually work hand in hand. And that's a little understood phenomenon. And the third thing is, because we use these cells as a model of what happens in the embryo, I have a collaborator, Margot Day, who works just upstairs in fact, and what we are trying to do is take the molecular mechanisms that we've learned from stem cells and see if they actually apply to real embryos. Because it's a model, you would expect it to, but it doesn't necessarily happen that way. But in general, all of the information we've gathered from the stem cells and have applied to the embryos themselves have strong analogies. The mechanisms are the same or very similar, so that we're showing that that the stem cells really are a useful model. Now that's of value because there's a lot of IVF goes on in Australia. One of the things you have to do is grow small embryos in a dish out to an early stage before they get implanted. That's not a very efficient process. So if we can understand the molecular mechanisms of early development, we can improve the culture conditions for those embryos with the hope of improving the success of IVF.
And you start with mouse embryos?
We work on mouse embryos, that's right, because it's easier to do, and ethically it's very easy to do. It's very hard to get hold of human embryos. So we're using the mouse as a model for human development, which is a reasonable thing to do. Again, whatever mechanisms we find in the way mouse embryos develop are likely to have very similar parallels or exactly the same mechanisms in human embryos. We are starting to work closely with a company that will help us actually look at human embryos to see how strong those parallels are, and to see whether what information we're learning from the mouse embryos if it really does help to improve the development of human embryos in culture before they get reimplanted.
How would they be able to do that?
In Australia, there are embryos that are frozen away that might never be reimplanted into the womb of the recipient, hopeful mother. They can either be thrown away or they can be used for further experimental research if the ethical clearance is there to do that. Some of those embryos are really early embryos, like 1 or 2 cell embryos. So if we learn how to improve the growth of a mouse embryo in culture, then we can try and get hold of some of those early embryos and see if that helps to improve the development of human embryos in culture.
So there's hope there for helping developmental illnesses that happen in the womb as well as inherited diseases.
I guess that's one of the possible useful side effects, if you like. So let me go back a step.
Sure.
For IVF, It's a reasonably inefficient process, but it does work. There are some problems that IVF babies tend to have more often than normal birth babies, including things like cardiovascular increases in cardiovascular disease, etc. Some of that may be due to just poor early development. In other words, very long-term effects out 20, 30, 40 years might be due to poor development in the first few days. So if we can improve the quality of development in the first few days, that may assist in reducing long-term effects for some of those children.
So the IVF babies will benefit from these techniques being used to produce healthier embryos and therefore healthier people?
Possibly. But there are advantages earlier in the process. IVF is about 20 to 25% efficient. It's not very efficient. So if you can boost that, that's good. It means that you can reimplant fewer embryos with the hope of having some success. And actually that figure of 20 to 25% is a little bit disingenuous, because that figure is only for the embryos that are reimplanted. What you don't get in that story is the embryos that never make it to the stage of being implanted. And that's quite inefficient as well. So all up, it's a very inefficient process. It's a very expensive process. It's a time-consuming and very fraught process. So if there are ways and means of improving the efficiency of that, there are benefits all round.
So you're doing fundamental science on the development of embryos, and so there's unlimited possible future benefits. You can't always tell because you're doing the fundamental science that everything else is built on.
Mm-hmm. That's right, but it's certainly not the only way to go. You probably know yourself, there are any number of ways now, large number of ways, of, for example, trying to understand the molecular mechanisms of development. You can do it with embryonic stem cells, you can do it with adult stem cells, you can do it with induced pluripotent stem cells, you can do it with eggs and sperm. So in order to understand the fundamental processes of development, using embryonic stem cells is one of the ways to go. In terms of producing cell therapies, embryonic stem cells is one of the ways to go. It may be that a cure for a particular disease or injury could come from using embryonic stem cells as the original source of cells, or it could come from using adult stem cells, or it could come from using induced pluripotent stem cells, or whatever. I think the important thing is that you need to keep all of those options open, because nobody knows which source of cells is going to be the winner in terms of developing an effective for the patient treatment and cost-effective treatment. There's fundamental issues with respect to the ethics of using cell therapies, particularly for embryonic stem cells. And that's been a debate that's been raging across the world, really, ever since human embryonic stem cells were first isolated in 1998. There's no problem with having those ethical debates. And in Australia there's been vigorous debate which has resulted in very directed legislation over the years, where the legislation is revisited to see if it's we're on course. And I think in Australia we're doing that job pretty well. At the moment, for example, if you want to make a new embryonic stem cell line from humans, it has to be frozen embryos made for the purposes of IVF that are no longer going to be used, and probably their development wasn't that good, so they wouldn't have been reimplanted into the mother anyway. And it has to be with consent, and there's no financial gain either from the healthcare workers or for the donor. So all of that came about through robust debate over the years, and I think we've got the settings about right in this country. In other countries, it's more liberal, much more liberal. In others, it's extremely tight. But I think in terms of getting the research moving in an ethical way, we've done a pretty good job here.
Michael Morris, thank you very much.
It's a pleasure. Thank you.
That was Michael Morris, head of the Embryonic Stem Cell Laboratory at the Bosch Institute at the University of Sydney, investigating the basic science of embryology to find out how stem cells turn into other cells.
Are you a scientist, artist, biohacker or maker who'd like to be interviewed about your work? Send your contributions, opinions, helpful suggestions and donations to science@diffusionradio.com. That's science@diffusionradio.com. Please rate the show on iTunes and Spotify. Tell your friends. Follow me on Instagram, Twitter and Mastodon @Ian Woolf. The news music was Rhinos theme by Kevin MacLeod of Incompetech.com. I produce Diffusion, which is broadcast around Australia to 28 stations on the Community Radio Network, including Tune FM at the University of Armidale in New South Wales, 2MVR in Nambucca Valley, 3MBR in the Mellie-Border districts of Victoria and South Australia, 4DDD in Dalby and the Darling Downs in Queensland, 4RPH in Brisbane, Denmark FM in Western Australia, 6GME in Goolarri in Broome, Radio Adelaide in South Australia, Lofty FM in the Adelaide Hills, 2XX FM in Canberra, City Park Radio 7LTN in Launceston, Tasmania, and Print Radio in Hobart. Diffusion is narrowcast on Indigo FM 88 in Northeast Victoria. Subscribe to the podcast on the Diffusion website, www.diffusionradio.com. That's www.diffusionradio.com, and check the website for links, photos and videos about this week's show. If you enjoyed this show, you can explore more than 1,300 previous episodes archived on diffusionradio.com, where the shows are labelled by keywords, so you can focus in on the stories you want to hear. I'm Ian Woolf. Join us inside your audio device of choice for more science wondering next week on Diffusion Science Radio.
Science is fun. It helps you to learn, to know, and to appreciate. When you study science, you may go on field trips. You discover the marvelous interrelationships between all living things. You learn to read the history of the Earth as it is written in rocks and fossils. You find out what makes things tick. Everything from a molecule to a living organism. In the study of science is found the most useful and satisfying knowledge of man. Knowledge of his physical world, its past, its present, and its future. And in your moments of relaxation, now and in the years to come, you will find the study of science leading you into fascinating pursuits. Photography, collecting. Why study science? Study science because you will find in the study of science a richer, more rewarding life.